the next speaker is Joanna Kirkpatrick from Thermo Scientific. If you could come up, thank you. Hi, thanks everyone for the invitation to speak here. I've got 10 minutes to talk to you about some of our new proteomics workflows from Thermo Fisher. So it's called Velocity DIA. It's enhancing the data acquisition and data evaluation on Orbitrap-based research. And if I can whistle stop through my talk, then we'll talk about wide window acquisition and chimeras if I've got time. So yeah, why do we want to quantify proteins? Well, it's great to identify them, but if we can add quantitative information, then we really make all the difference in terms of looking at something that's biologically interesting. It gives us confident characterization to really measure the differences in biological systems. And for that, we need to be very accurate, we need to be precise, and we need high throughput, especially when we're talking about a clinical environment. So there are many different choices. Every method has its pros and cons, if you like, and you have a fit-for-purpose assay. So the most accurate and precise would be a targeted measurement, but this limits you to the number of analytes that you can look at. We can go to multiplexing, which lets you look at more samples in one go, but tends to have some systematic errors. We can go label-free quantification, which is on the mass spectrometer probably the easiest thing to set up, but then we have slightly less precision. And if we go for low resolution instruments, then we have very low mass accuracy, and this is then not necessarily always well suited to quantification. So we have all these different options up here. TMT quantification is something that we've been doing for a long time, and it's great when you've got a few samples that you want to really directly compare to each other in the same analysis. But this tends to limit you slightly in terms of your how many you can do in terms of your throughput. And so what the field has been doing lately, and probably everyone's heard, is this data-independent acquisition. With high resolution, this allows you to increase your sample cohort size and make many, many more measurements in a high throughput fashion. So why do we want to do this? Why do we want to do data-independent acquisition? So historically, mass spectrometry was all data-dependent acquisition, and this could be done in a label-free fashion as well. The problem with DDA was that it tended to be stochastic, so when you increased your sample cohort size, you ended up with a lot of these things which we call the missing value problem. And for large sample cohorts that you need to measure in a clinical study, we need to measure lots of samples, and we need to make sure that there's as much data completeness that we can achieve. And then we need confident, reproducible identification and quantification at the same time. And we really need to keep this missing value problem down as much as possible. And so DIA tends to be called like all of the data all of the time. So you're actually, rather than targeting anything, you're just scanning through the mass range and acquiring enough MS1 and MS2 for quantification whilst identifying everything. Data is very complex, but we're really lucky that a lot of the data analysis software tools now have really enabled us to deconvolute the spectra that we achieve. So we need to get the high throughput in order to be able to measure enough samples, but we also need to maintain that accurate quantification of as many proteins as we can. So yeah, just to very quickly explain data-independent acquisition, it's a very fine balance between maximizing the number of protein identifications whilst maintaining the quantification accuracy and the throughput. So all of the ions within a certain mass range are fragmented at the same time, and then we have to deconvolute that spectra afterwards. So it's really important to consider all of these things when designing your method: the isolation width in MS1 that you choose, what mass range you cover in MS1, what resolution you measure your DIA, how much you overlap your windows and collision energies. These can all influence how fast you can go, the depth of the coverage of the proteome that you can achieve, and the quantification accuracy. And so, yeah, if we go for higher throughput with more IDs, we have to reduce certain things, or if we put the resolution up, we can get higher quantification accuracy and higher precision, but we might sacrifice a few identifications. Window width will make, obviously, things go faster if we make that wider, but then we potentially have a bit more challenge in our precision and everything else. And obviously, the mass range that we cover is going to limit everything, because we have to go through the entire MS1 mass range for each of the DIA scans, and that can also have an influence. So we've come up with our solution to doing this in a high throughput fashion, because yeah, DIA has been around for a long time, but really getting it to go a lot faster has been one of the biggest challenges. So we've come up with this workflow we call Velocity DIA. for label-free quantification of the proteome. So that includes at the moment our new Vanquish Neo HPLC. Very importantly, these micropack Neo columns, which we'll talk about a little bit more in a second. Ideally, the easy spray source in front of either our Exploris 240 or 480 instruments. And then, as I explained, data analysis is also really important. So at the moment, we're recommending Biognosys from Spectronaut from Biognosys as the industry standard for processing DIA data. And then this really gives us the best compromise, if you like, between throughput, quantification, and also gaining as many identifications as possible whilst maintaining that quantification accuracy. So we have then a reproducible and robust and consistent solution for then the large cohort studies. So yeah, this is a bit more detail about the micropack columns and why it's so important. So peak shape is very important when doing DIA because we're talking about measuring our quantification in MS2. And you can just see there on the video the difference between a standard packed column and these micropillar columns. If you want to talk more about in detail with this with anyone later, Robert is somewhere around and he will talk to you forever about micropack columns and how amazing they are. So I won't go into any more detail, but yeah, this is really important for the DIA because we need really reproducible chromatography. And so we've obviously benchmark this on some gold standard ground truth data, and we do this 3-proteome mix where we mix yeast, E. coli, and human in different ratios, and we have a very high background of the human proteome, and then we mix in different amounts of yeast and E. coli on top, and then we optimize the gradient for 30-minute runs, and this was actually done with Spectronaut 16. There's now Spectronaut 17, which does an even better job because they've improved a lot of the algorithms. And we really looked at the data to see how much we could make this faster by picking a very particular portion of the MS1 mass range. And you get 90% of the identified peptides in a mass range of 400 to 900 m/z. So this is what we choose in order to maintain that speed. And yeah, then also, probably slightly differently from your classical proteome experiment, we are going a little bit faster with the chromatography to really make the most of all of the runtime. Is it going to go backwards? Can I go backwards? Yes. So, sorry. Yes, to make the most of all of the runtime. We have speeded up the chromatography so that everything that you get really in the 30 minutes, there's peptides in your chromatogram. And these are just all the settings which we can talk to you in more detail about if you're interested in the finer detail of how we've achieved this. You can come and talk to Richard or myself on the booth later. But what you can really see here is that we have very good quantitative accuracy for the known ratios that we spiked those different species proteins into each other. So you can see here, you can see here on the left that we have very, very good quantification accuracy of all the 3 species and the different ratios that we spiked in. So yeah, to summarize, we've got this optimized high-resolution DIA on Exploris 240 or 480, which is the right combination for getting your throughput, your quantification, your robustness, and the identifications that you need to be able to measure large sample cohorts with the good depth of coverage, whilst maintaining all of those other really important factors for the quantitative analysis. So to summarize, this is a combination of the Vanquish Neo micropack columns, the Orbitrap Exploris, and it will give you the ideal workflow for high throughput and label-free proteomics and quantification using data-independent acquisition. We've got the short 30-minute gradient, which really lets you get through a lot of samples in a day. All of the great software tools that are provided by Biognosys that let you do direct DIA, so there's no need anymore to make a library. Gives you really good proteome coverage. And the DIA data is really high quality, which leads to excellent quantification, which is what you need when you're starting to look at the biological questions that you really want to answer. So we have very good median CVs of the quantified peptides, which is really, really important when you're starting to look at the things that are interesting. Right, I have 30 seconds, according to my timer, just to introduce chimeris and wide window acquisition. So sometimes DIA is not necessarily the best solution, and specifically in the single cell world, your spectrum and your chromatogram may not always actually have peaks because you're dealing with such little amount of material. And so what's been developed lately is like the compromise between DDA and DIA. And so it uses the DDA to trigger an acquisition, but on a wider window, so that you still get chimeric spectra, but you get it where there's actually signal in your MS1. You can sacrifice some of the speed of your acquisition because you have less signal, which means you can really optimize the resolution for your MS2 and get very complex MS2 spectra. But we have this new software called Chimeris, which can really deal very well with deconvoluting that very complex spectra when you've got a high resolution MS2. And the single cell field is really thinking that this is quite exciting at the moment because it's providing this perfect hybrid between DDA and DIA for very low level data. I am now 30 seconds over, so I'm going to skip how it works. Come and talk to us at the booth if you would like, because don't have time to go into too much detail anymore. But basically, the take-home message is that it can allow you to do quantification at much lower level than we have with any of the other solutions at the moment. And this is just an acknowledgement of everyone who's contributed towards the slides I just presented and the data that I show you. Yeah, so please come and talk to Richard or myself if you want to know more about the DIA or the Chimeris wide window acquisition. Thank you.